Hello everyone, this is Alexis McAleer presenting exam mini video 3 prompt 2 on synapsis of chromosomes. To start, meiosis occurs where gametes known as sex cells are formed due to cell division. During meiosis, cells undergo prophase 1 where homologous chromosomes begin to pair up. Synapsis then occurs while still in prophase 1. Synapsis is a process of sharing genetic material between the cells. Chromosomes use special techniques to fuse together where chromatids overlap in order to exchange their genetic material. Here are the different chromosome pairs in both individuals in the study. The black dots between the loci depict the centromere. Here's individual 1. Individual 1 undergoes pericentric inversion. Not all of the loci are involved, only I, J, and K. The inversion causes compensation because of the looping. This is due to trying to align the loci. Not all of the outcomes are viable, only 2 of the 4 or 50%. 2 of the outcomes are not viable because there are duplications in the loci. There is also a crossover between J and K, and gametes produced are listed to the right. In individual 1, we saw compensation because there were duplications of the loci. Here in individual 2, no compensation occurs because the chromosomes are identical. There is, however, a crossover between J and K. All outcomes here produced are the same. That is with a 100% viability rate. When looking at the differences between individual 1 and individual 2, we notice that the main difference is the rate of viable offspring. Individual 2 has a rate of 100% viable offspring versus individual 1 that only has 50% offspring. Individual 1 underwent pericentric inversion, which had compensation due to the looping, and then their offspring had duplicate loci that made them not viable. Therefore, individual 2 would have a higher fitness. Here are my references.